Previous studies found that it's possible to remove the cells out of mammalian tissues and plant material. Furthermore, it was possible to seed culture cells onto the remaining scaffolds. On both scaffolds obtained from plant material and mammalian organs, the cells survived. The aim of this project was to both reconstruct what was done in the past, but more importantly to advance the culturing of the cells in and on plant leaf-based scaffolds in a bioreactor. The plant leaves were purchased at the local grocery store. Storing the leaves in the refrigerator kept them usable for at least 1 week. In order to compare the leaves of different plant species in terms of their efficiency during the decellularization process, multiple leaves were decellularized simultaneously. Basil, parsley, and spinach were selected for their use in previous studies. Cannulas were used to later on infuse liquids into the vein system of the leaf. The cannulas were sanded and fixed to the stalk with dental glue. After washing the leaves with distilled water, the leaves were hung up with the petiole pointing upward. A 10% SDS solution was infused into the leaf stalk. Throughout the treatment with the SDS solutions, the leaves were surrounded with distilled water in order to prevent dehydration. In order to remove any remaining proteins and green color, a mixture of an aqueous sodium hypochlorite solution containing 1% of the surfactant Triton X100 was injected into the leaf. When the decellularized leaves came in contact with water, at first a solvable white substance was settling down inside the leaf's vein structure. It was removed with distilled water over time. After the leaf scaffolds were coated with a 1-to-1 mixture of FBS and PBS, the cells were seeded by either injecting them into the vein system or simply pipetting the cell suspension on top of the leaf. Human umbilical vein endothelial cells (UVECs) were used to inject into the leaf, and L929 cells, a mouse fibroblast cell line, was used to seed the cells on top of the leaf. The efficiency of decellularization was the best with spinach leaves. A direct comparison of parsley leaves and spinach leaves, which were treated the same, showed that most of the spinach leaves were free of green color and had no remains after washing. Under the same conditions, parsley leaves needed 7 more days to completely decellularize in comparison with spinach. Referring to the decellularization, basil leaves showed a similar performance as spinach leaves did. Basil leaves were harder to infuse with cannulas due to their thin stalk. Therefore, for further experiments, mostly spinach leaves were used. According to literature, one of the major components of the extracellular matrix of plant leaves are cellulose microfibers, which create a highly polar environment for the seeded cells. In theory, cells, especially fibroblasts, should be able to adhere to the plant scaffold without any surface functionalization. To investigate that, cells were seeded onto basil leaves, one with FBS coating, the other one without. The fluorescence images show that coating the scaffolds with FBS makes sense. The uncoated samples had only a few adhered cells, in contrary to the coated leaf where the cells, interestingly, adhered to the region of big veins. A comparison between injecting the cells and placing them on top of the leaf was not possible using 2D fluorescence imaging in combination with Z-stacks on UVECs stained only with the DIL dye. In order to locate the cells in a 3-dimensional room, the staining of a cross-section or the use of a 3D imaging method, such as laser scanning microscopy, is suggested. Designing the bioreactor was successful. The medium reservoir, as well as the incubation chamber, were bottles used for solvents in the HPLC. In order to create a constant stream of fresh medium, the medium reservoir was elevated, so a hydrostatic pressure was applied. Sterile filters were used to keep the incoming air, as well as the medium, sterile. The apparatus ran successfully for nearly 2 weeks with cells inside. 2 bottles, each 200 ml of medium, were consumed. Unfortunately, no cells adhered, most probably because the cells were injected inside the bioreactor and the medium was already starting to flow after 30 minutes. For the future, improvements could be to inject the cells horizontally, as they were in the experiments before, increase the delay between seeding and starting the flow, and as a final improvement, consider using a pump to apply the medium flow. A pump would allow to control the flow rate way more precise than with the hydrostatic pressure.